So welcome to today's session on emerging topics in biomolecular NMR spectroscopy. My name is Art Palmer on behalf of the other organizers. I welcome the participants today and welcome our two lecturers today. You see on your screen our front page for this lecture series. There are two important pieces of information. The first is that we follow the Gordon Research Conference policy. Please do not make audio or video recordings of the lectures by any means. With speakers' consent, we record the lectures and then they will be made available at the ICMRBS YouTube channel. The website for the YouTube channel is shown on the screen, but you can also find that information by going to the website for this lecture series, emergingmagneticresonance.org, as you see at the top of the screen. With that short introduction, let me turn to our first speaker today, Anja Böckmann, who is in the Institute for Molecular Microbiology and Structural Biochemistry, a joint institute of CNRS and University of Lyon in France. I knew Anja's work when she was a graduate student in Paris doing solution NMR spectroscopy, and it was then a mixed pleasure when she moved to New York to do a postdoc in solid state NMR with my colleague Ann McDermott at Columbia. It was terrific. I got to know Anja and learned quite a bit from her actually over the years. But of course, solution NMR spectroscopy lost a spectacular spectroscopist over to solid state NMR. After her postdoc, she moved back to Europe. And as I said, she now has her research group at the University of Lyon with a continued interest in both methods development and applications of solid state NMR to very difficult biological problems, including fibrillar proteins and membrane proteins. And she's going to tell us about her work today. Anja, thank you very much for giving today's talk. And now you should be able to share your screen.
Okay, I hope this works.
All good so far.
Okay, so thank you very much, Art, for this kind introduction. And I'm sorry that I preferred in the end solid state NMR, but it's very exciting actually. So I'll report today on proton-detected solid-state NMR experiments on viral protein assemblies. And before I run out of time in the end, I would like to thank already my collaborators and my co-workers here. This is from my group, Marie-Laure Faugeron, Lauren Lecoq, Shishan Wang, and Marie Dujardin, who worked on this our project, and all contributions from all the group. Michael Nassal is a virologist at Freiburg University who is a very important collaborator on everything which is hepatitis B capsids. And then we have a collaboration from industry, from Gilead and Janssen on some compounds, which I'll talk about today a bit. And then this is a collaboration with ETH Zurich, with the group of Beat Meyer, and this is Susanne Penzel, Martin Schleder, and Alexander Malher and Beat on the spectroscopy side.
Anja, your slides aren't advancing if you were advancing them.
They aren't advancing? Okay.
No. And then while you think about that, I forgot to say something important, which is for the audience, type any questions you have into the Q&A. And then at the end of Anja's talk, I'll ask the questions. If you want to be unmuted, use the raise your hand function if you want to ask a question directly.
No, this does not work neither. So I have to get rid of the screen. And is that okay?
We see the front, now they're advancing.
Okay, so you didn't see the slide, but you'll see it again in the end, and you heard the text with it. So I'll just go forward. So I'll talk about hepatitis B virus infection. And this is actually another viral infection. Everybody knows today much more about viral infection than we knew 1 year ago still. So hepatitis B is actually another virus, which is a huge problem today in the world. And actually 257 million people have lived today with chronic hepatitis C infection. Actually 1/3 of the population of the world has been infected at some point with hepatitis B. So the people who have chronic hepatitis B is about 10 times more than coronavirus. There's every year about 800 to 9. So this is, you can compare that now to COVID-19. This is about the same number, but this for hepatitis B, you see in the map that this is mainly concentrated in Africa. So it's really a huge problem there. You see that all the developed countries are doing pretty well. So there's a vaccine available. This is because of that. Everybody is vaccinated in Germany, for example, in France also, US, and this is available and efficient. The treatment, if you have it, if you need it, is daily and lifelong. So to access that is more difficult, and there's still no cure. So there are really new treatments needed. And a big problem also of this virus is that perinatal transmission is most common, and chronic hepatitis B in infants is very deadly. This is also reflected in this paper in Nature 2 years ago. So hepatitis B is actually killing more people today than HIV, malaria, or TB. So we need to make an effort on that, and we would like to contribute. We work mainly on the hepatitis B viral particle. It's a very interesting virus also. It's one of the smallest viruses, mammalian viruses. And you can see here the assembly of the viral particle, which is not done in one step, but it takes several steps. So you have the core protein dimers, which are the building blocks, and they assemble to a first kind of capsid where together with the polymerase and also with the pregenomic RNA you see here. So this is the immature capsid. This then matures, and maturation here means that the RNA is transcribed to single-stranded DNA first, and then to partly double-stranded DNA. And there, at some point, the capsid has to change shape, because in some way, which is not well understood today, it signals that it's ready for envelopment. And then the surface protein, there are 3 different ones. You see the small one, the medium one, and the large one in yellow here, with these little extensions for the medium and large one, because they have an N-terminal addition. And they then assemble the particle and also interact with some cellular proteins. You see here a chaperone, which holds this N-terminal to the inside on assembly. And this then goes to the outside somehow, not well understood neither, in order to interact with the cellular receptor, which is the NTCP here. Today, I will focus on a new approach. It's a new class of HBV antiviral, which is under development today, which are capsid assembly modulators. So they act on the assembly step of capsids. And you see this step here a bit drowned in the middle of the viral life cycle, which I don't want to bother you with. You just should look at the red rectangle here, where you have then the pgRNA, the pregenomic, which is packaged by the core protein dimers to become the capsid. And it's this step which can be addressed by capsid assembly modulators. You can see here the structures. There are actually two types of these assembly modulators. They're all small molecules. They are mostly hydrophobic because they interact in the hydrophobic cavity, which is located at the interdimer interface of the capsid proteins, which I will show you later. And so this interferes with this packaging step. And you have 2 classes, and the one class actually, I'll show this here. The dimers, when you add RNA, so they assemble to the nucleocapsid, and when you have then these assembly modulators, the type 1, you get apparently normal capsid assemblies. And when you have the type 2 ones, you get aberrant capsids. So they really perturb the assembly. The apparently normal ones, actually, they are believed to form much more rapidly, and by this, not allowed to package the RNA and act through this way. So to study that is one point easy, but the other one is difficult. What is easy is to do that on truncated capsids, which do not pack RNA, which are modeled in the lab. But it's very difficult to start to study this assembly in vitro from full-length protein dimers. This is from protein which packages RNA and also in the presence of RNA. So we thought that we can investigate actually this process with a system we have in hand, which allows us actually to study that directly on exit of the ribosome of the capsid proteins. What will we need for that? Actually, the sample we studied at the beginning and which we also continue to study come from bacterial expression. Michael Nassal has optimized that, and we get large quantities of the capsids. And actually they assemble already in the E. coli cells, and then they can be disassembled also under denaturing conditions and be reassembled. But we would like to do this by self-resynthesis actually, because this is an open system, so you can add capsid assembly modulators directly on synthesis. So you can observe, or what you can see the impact of assembly modulation directly at the exit of the ribosome with the system. However, it's rather well known that self-resynthesis delivers small quantities, and what I mean with small is below the milligram, but so 0.5 milligram to a milligram is easily obtained. So we need to change also our NMR approach, because we work most often with C13 detection still, which needs large quantities, but often we have them since the E. coli they produce them. C13 detection has a low mass sensitivity, what you know from solution NMR also, where mostly proton detection is used. And you probably know also that solid state NMR can use proton detection with going to faster and faster magic angle spinning rates. And the important advantage in this context is that proton detection allows us to detect the small quantities or to work with the small quantities we get from self-resynthesis. So first I'll talk about the proton assignments of the HBV capsid. So to do this already, I'll just say something about the protein also. So here you see the real HBV capsid. It's an X-ray structure which has been obtained already 20 years ago. This is the truncated version, which is called CP149. And you can see that there are 4 different molecules in the asymmetric unit in this structure. So one type forms the pentamer interfaces you see here in green, and the 3 other types, they form the hexamer interfaces, and they all form dimers. There's only one monomer color colored here. So the dimer has the structure. You see the spike helices here, which are composed of the interface here between the 2 dimers. And you can see here the organization of the sequence. So the assembly domain is from residue 1 to 149. And then you have the C-terminal domain, which is essentially responsible for the RNA interaction. And you see that there are many arginines here. And also that you have several phosphorylation sites here, which are very important also to direct the capsid maturation and also afterwards interaction with the nuclear import factors and so on. So as I told you, we started with carbon 13. So this protein or these assemblies, these are entire capsids, which are just centrifuged into the rotor. So they give a very nice spectra. We did assignments using 3D experiments, classically NCACB, NCACX, CANCO, and NCOCX. So this was a rather straightforward action. And now I would like to go to proton detection. So we used for that a series of experiments to see also whether we could, in principle, de novo assign that. So this is HNCANH to one side, HNCONH to the other side, and then you jump the CO also in one experiment. And then there's a 4th experiment, which is a bit more complicated, many transfers, where you get the information on the C beta also. I won't go into detail on the pulses there, but you can see that they mainly use still dipolar transfer steps. The last one also uses scalar transfers, but the dipolar ones generally work better still in MIS. This is, has been done at 100 kilohertz. mass. So you can see here some extracts from the spectra. The different color coding is indicated here. And this is a short stretch where you can see already that the signal to noise is rather nice for the red, the green, and the cyan one. And the violet ones, there are not so many signals because you have too many transfer steps, and this is getting difficult. Still, we could assign all the NHs of that. I told you in principle we tried to make that de novo, but obviously we had already the N15, C13 assignments, so we checked that and it was coherent, and we established the NH resonances. So just to say something about there was an occasion for us also to compare really what C13 detection and proton detection, where it does make a difference. You can see here the spinning speeds used. So the proton detection was done at fast MAS in 0.7 millimeter rotors. The quoting quantity is here about 30 milligrams, and it goes down here, as I said, to about 0.5 milligram, which is very advantageous. Labeling schemes, as you'll see later, we tried here also deuterated proteins and also protonated proteins in the proton detection. The temperature is a bit higher in proton detection because you have to counteract the fast mass, which is not always so easy. The typical fingerprint experiments are in carbon-13 detection, the DAR or the NCA, and in the proton detection, the HNH. The experimental time for that is about the same. And also the 3D experiments we recorded, there are 4 of them which are more or less fulfill the same functions. The total experimental time was also very similar. And as you can see, the backbone assignment was also very similar. So the biggest difference is actually this factor of about 100 where you go, which you lose in the need of sample, which is a very good thing for us because we start to work really on proteins which are only available in small quantities. Just to say something more about sensitivity, so you can see that the signal to noise in the proton detected spectra was a bit lower. So in the end, you gain more or less a factor of 50 in mass sensitivity, which is still very good, I think. The assignment completeness, you see that there's some residues missing from the proton spectra, which we couldn't assign. That was mainly due to overlap, but it's rather complete also. One interesting thing is also, I told 4 different molecules in the asymmetric unit, and you get for a set of peaks, you get really several peaks also, because the structure is not the same of the 4 molecules. It's not for all the signals, but it's about for 20% of the signals. And you can see here for alanine 137 here, for example, the NCA correlation spectrum, where you can see here the 4 peaks of the 4 different monomers. And you can see here the same thing or the equivalent region in the NH spectrum. You can see that the dispersion in the proton dimension really adds to be able to distinguish these different forms from one another, where in the C-alpha chemical shifts are more or less the same, and they do not resolve these structural differences. So the proton chemical shifts, they really add something here in order to separate these signals. Does deuteration provide an advantage still? There's a lot of discussion, and people sometimes say at 100 kilohertz you don't need to be deuterated anymore. It's true, you can work on protonated proteins, fully protonated, so this is 150 amino acid protein, and you get a decent resolution here, but you still get a better resolution when you deuterate. You can see that here for this isolated peak, and I found that impressive also to see it on the FID, that you really have a signal which lasts quite some time longer. And we did quantify that then, measured relaxation time. So this is bulk T2 prime, which gives the coherent line width. And you see that this is still the difference between the deuterated and the protonated samples. You have here about 2.5 milliseconds, and here more than 10 milliseconds of the signal lasts. Is still dominated by coherent contributions, so you get here really a narrow line due to the dilution of the proton coupling network. We also measured that in the site-specific manner. You see that on average, you get on the protonated sample 170 Hertz line widths, and on the deuterate, you lose quite some, which is a good thing. You go down to 110 kilohertz. So the lines are about 60 Hertz narrow on average, which is very useful. We also wanted to see what this gives if you go to even higher magic angle spinning frequencies. So this is at 150 kilohertz MAS. So this is with a 0.5 with a 5 millimeter, no, 0.5. So this is fully protonated CP149. And you can see actually that the homogeneous contribution is getting really small. It's a bulk average of 86 Hertz line. But heterogeneous contribution, which in the capsid can be a bit bigger also than in other proteins, because you have these 4 different molecules, and even if in the spike tip you don't see much difference anymore, this can just contribute to the line width because it's not resolved actually, the chemical shift difference. You can see that also we measured that on different proteins. So this is Het S, RPO, and in yellow you see protonated, so they're all protonated CP149, and you can see here when you go from 100 kilohertz to 150 kilohertz that you gain about 40 hertz in the line widths of the bulk the bulk signal. So faster MAS is useful. The faster we can get, I think it's the better. There's still signal in there, and if the line widths they go more down, then we also gain more in signal to noise. I'll say another thing about deuteration. In E. coli produced proteins, it actually gets sometimes difficult, and we can see that in the HBV capsid, to back exchange all the protons, because in E. coli you make the protein in D2O first, and then you have to back exchange the protons, and if they're hidden in the hydrophobic core, then this can be difficult. And we see that here. You see these red dots here where we do not see a signal in the deuterated protein here. So they're not back exchanged, and that might be a problem for large proteins, which you would like to study with this method. But when you have even more resonances, then you need the deuteration. And this can be complicated to really obtain all the NH signals without denaturing and renaturing, which can be very, very complicated to hopeless in large membrane proteins, for example. So the second aspect, we need wheat germ cell-free protein synthesis of the HBV capsid in order to add the CAMs directly to the protein on exit from the ribosome. We would like to put that into an NMR rotor and the spectrometer, and then record these spectra in presence of CAMs. In the lab, we have set up in the last years a not so common cell-free protein synthesis approach, which uses wheat germs actually. This is as plants. They have the eukaryotic ribosome actually, which synthesizes the proteins more slowly, and which normally helps to better fold them on synthesis already. And you get, notably for eukaryotic proteins, this can really make a difference. So we make these extracts. We add the mRNA for the protein of interest. And then we here use that using dialysis cups in a solution, in a buffer solution. So you can add here, obviously you add labeled amino acids. You also add some energy producing or providing substitutes, and you can add the CAMs, and then you get this isotopically labeled protein sample, and you can do that for several proteins. We tested that both on the truncated construct, because this one is often easier to work with, and also on the full-length construct. And actually we have seen that the truncated capsids here, despite that they have the entire assembly domain, they do not assemble in this approach. So you can, we produce these proteins in the cell-free system, we separate the pellet and the supernatant, and then we load the supernatant on the sucrose gradient to make a purification step. And you can see that the truncated capsids, they don't even enter the sucrose gradient. And you see in the electron microscopy picture that they do not assemble. You see some few capsids, but there are not many of them. And when we use the full-length protein, which does assembles around the RNA, which is present in the reaction, you see then that they assemble. You see all these capsids under the microscope, and they migrate to the 50 and 60% fraction. We made a labeled sample then, so this was deuterated. You can see that on this one we were lucky because it's rather poor. Sometimes we have more contaminants. This depends a bit on the student and on the setup used. Labeled samples can be obtained just by addition of the labeled amino acids to the reaction mixture. So this is a very nice thing also, that if you have contaminants, actually you do not see them because they are not labeled. So these samples are purified by this simple sucrose gradient, and then they are centrifuged into the NMR rotor. And here, just to show you a comparison, we have made protonated cell-free CP183 capsids, and we have made the deuterated equivalent also. And I compare this here to the protonated samples we got from E. coli. And you see that the spectra are also nicely resolved. They took a bit longer to record, but even not so much. You can see that here, where I compare the signal to noise per number of scans of the E. coli and the cell-free capsids. For the protonated version, either the rotor was not very full or we had more contaminants. This is not very clear. We got less signal, but for the deuterated sample, it was really nice expression, and we got even more signal to noise on the rotor than from the E. coli sample. On this one, we could also record a 3D spectrum here. You see it's not a projection, but it's showing all planes overlaid on the 2D spectrum. And you can see that the line widths in the cell-free samples, they were comparable to what we obtained also in the E. coli samples. Another nice thing in cell-free expression is that as the reaction takes place in H2O, you have no need to back exchange in deuterated samples. So you get directly protons on the exchangeable sites. I just put up this picture again from before, where you see that in E. coli we miss several resonances, and actually you can see them here in the spectra of the cell-free protein. They come very nicely, and we were able to find them here also. So this is a good thing. So now coming to assembly modulation, we have until now the proof of principle, and I'll show you in the next slide the first spectra we have also, but these were the experiments which made us to go further. So this is cell-free expression, and you see here the Western blots mainly, that in presence of DMSO, the protein still is expressed because we need DMSO to dissolve these hydrophobic molecules. You can see that this works quite nicely still. And then we added 3 different types of assembly modulators, 2 type 1s. This is AT130. It's rather historic molecule, so there are much better options than this one now. Whereas, for example, this here is a newer molecule. This is also a type 1. So you can see for these you have assembly of the capsids in the reaction. This here is a type 2 CAM, and you see that in the E. coli they build these open objects, and in the cell-free reaction you can observe also these open capsids. So the principle works, and we recorded first spectra. You can see here overlain on the CP108 capsid without CAMs, so we have 2 different ones here. And we observe large CSPs from the binding of the CAMs. So you can see here, for example, peak A moves quite a lot on the scale here. It's about half a ppm in protons, and even more than a ppm in the nitrogen dimension. Peak B also moves quite a lot. So this is where we stand at the moment. We have to now record 3D spectra also to assign these signals and to follow the chemical shift changes, and also to explore other things. We have, for example, observed that phosphorylation plays also a role in this process. This is also known from in vivo experiments already. the cell-free system can phosphorylate, and it does phosphorylate the capsid under certain conditions, and we'll go further into this also. Just to summarize, we have done NMR spectroscopy on the capsids. They yield nice spectra. Deuteration is helpful still and can be done in cell-free or in E. coli expression, but better and more complete in cell-free. In the cell-free protein synthesis, the capsids auto-assemble, which is a very interesting tool now. The reprotonation is not needed, and they are easily purified. And now we can go ahead and use the cell-free setup to investigate assembly modulation on wild-type full-length proteins in presence of RNA. So I would like to thank you very much for your attention, and just put back this slide. And if you have questions, please go ahead.
Thank you, Anja. And again, if you have questions, type them into the Q&A. Don't use the chat. And then I will ask Anja the questions. And again, raise your hand if you want to be unmuted. So first question, Anja, how feasible is it to further reduce rotor size and increase MAS frequency?
Well, you should ask Argo about that, or Broker maybe. But I think it is feasible, and probably it will help us to go further also. I mean, at some point, what people say at the moment is that the rotor gets so small that you don't have protein anymore in it, and you have to have the gain in line widths, which counterbalances that the loss of protein. And so 100 kilohertz for me was a good compromise at the moment, because you really start to have as narrow lines as you need to start working on a bit bigger proteins also. And you still have protein in the rotor, but as you can see at 150 kilohertz, we still get very nice spectra and lines. They get narrow again. So I think it will be very sample dependent, because as you saw that if you have too much heterogeneous broadening, then this will remain. This will not be spun out by the MAS. So I think it will be very sample dependent. It will not be good for all samples, but if we can get samples to have narrow lines, then we will profit again from faster MAS.
So I'm not going to go in direct order. I'm going to try to get some of the technical questions before the biology questions. So what is the largest scale or mass of protein you have performed with cell-free synthesis?
You mean the highest yield we get? Yes. Or the most protein we can produce? We normally produce what we need. So this is at the moment 0.5 milligram. But I mean, in principle, we do more or less 20 milliliter of cell-free extract per 2 months. So we do in a year 120 milliliter. If you count that, you can get about 1 milligram in nice situations per milliliter extract, then the limits is what you can do as an extract per year, I think, or per time scale. So this is this really depends on that. I mean, getting 30 milligrams of sample for 3.2 rotors for the capsid, we have more or less 50 rotors for the E. coli preparations. So that by cell-free would have been impossible, I think. Or I wouldn't have liked to have done it, because cell-free, wheat-germ cell-free needs this long step where you have to sort the embryos by eye. And this is the limiting step, more or less. But for E. coli cell-free, I think you can go ahead. I mean, you can do a lot of sample.
Oh, could you comment on side chain proton resolution at 150 kilohertz MAS, especially the methyl groups?
I didn't mainly think about methyl groups because they're already very nice. But what is really getting better is the CH2s. Because the chemical shift differences get better between the neighboring protons, and also the other, obviously, but these are the ones which are closest in chemical shift, and they profit the most. I mean, CH3s, everything gets better, but mainly the CH2s, they really, I mean, at 100 kilohertz, you see them, but you cannot resolve them.
So maybe you said it and I missed it. As a follow-up, what fraction of the side chains do you have assigned at this point?
The proton signals we did not assign any, but the HNs. So this is and it's not subject to interest at the moment. As I told you, the CH2s are, even at 100 kilohertz, they are At 150, Beat starts that with his students on this RPO, where in the latest paper he has they show the CH2 regions and it's getting better, but at the moment we don't plan to assign side chains. This is the nice thing with carbon spectroscopy, that this is really easily accessible side chain information. It's a big advantage still.
And Jeffrey asks whether DNP can help some more.
Well, maybe, but I'm always happy when I get narrow lines. So it's for me really hard to get back to times where lines were broad. So well, I think it's I think it's getting interesting actually when you use cell-free, that you can really label very individual sites in a protein, and then I think you can, if you have a good question, you can really address with a model where you can say either I see a peak here or I see a peak there, and I've only labeled one amino acid. I think then the DNP is extremely powerful.
So do you know what proportion of monomers end up in pentamers versus hexamers in the assembly?
Oh, yes, this is given by the icosahedra. So it's, I don't remember. It's, I think it's 60 to 108 to 120, if I remember right, or it's the same. No, I don't remember, but this is known. This is a regular icosahedra.
And does that match the relative peak intensities of the 4 alanine signals?
Not 100%.
Oh, Lauren wants to jump in.
Okay.
No, sorry, I was just trying to remove some of the questions from the list.
Ah, okay.
Once they're answered.
Go ahead, Anja.
So the peak intensities do not fit 100%. They are a bit stronger for one signal. So we suppose that that maybe the pentamer interfaces are more rigid, maybe something like that. Not clear, but it's about the same intensity, but it's not perfectly.
Let's see. Are you doing any NUS processing?
No, not yet. This is something Beat is developing, but I didn't use that yet.
Another technical question is, how's the temperature regulated in the 0.7 millimeter MAS probe?
The exact design, I don't know, but it's about, I mean, the gross design is a fridge, and then it's the same setup as for 3.2, but I cannot tell you the details.
So there's a question perhaps you'll understand better than I do. Have you tried observing a correlation between the proteins and the genetic material? And also, have you tried comparing the same proteins you showed in different growth stages of the virus?
No, I mean, the second question is no, because we do not look at the, I mean, this is very difficult to mimic. I mean, we try to make models which mimic these different states of the growth. For example, we looked at empty capsids and at RNA-filled capsids, but the real viruses and the real stages is for the moment impossible. Because we first don't have the security equipment neither, but also to prepare these samples is just at the moment impossible from real viruses. We try to mimic them, obviously, and we are starting on that with the easy accessible ones, and then go to the more complicated ones as the DNA containing ones and the envelope ones. So we work towards that. And the first question was, oh yeah, if we saw between the genome and the genome in the E. coli samples, as it's labeled also, but it's not the genome, it's E. coli RNA, which is packaged. And we do not see any correlations until now, because the C-terminal domain actually, which packages the genome, is largely invisible, and we are at the moment trying to explore, to use all possible NMR techniques to see if we cannot make it visible, and we are starting on to have results on that. So this is to come.
So do you see any evidence of heterogeneity in the capsid protein environment? Multiple signals for the same amide, for example, which seems to be implied by the irregular capsid shapes observed for type 2 CAMs?
No, it's actually the spectra with CAMs are better resolved than the ones without. So the microscopy images, they look as it would be horrible because it's there's no order, no long range order, no symmetry, but actually the spectra look very, very nice.
And when did they build all those churches in Lyon we see on your slide?
The slide is actually half Zurich, half Lyon. So they had two crews to build them. They started early in Lyon, so I don't know in Zurich, but in Lyon they started in the Middle Ages, so they had quite some time.
Okay, there's a few other questions in the Q&A, but I'll let you answer them directly in the Q&A so that we can move on to our second talk. Thank you again, and I'll turn control over to Oscar.
Thank you, Art.
Okay.
So it is my great pleasure to introduce Pramodh Vallurupalli. Pramodh is a great scientist and also an old friend from the good old times at Lucis's Scale Lab. So Pramodh started after his undergraduate studies. He joined the group of Peter Moore for doing the doctoral research. Peter Moore is one of the capital fellows in RNA and ribosome, where he did mostly structural studies. And then he moved to Toronto to do a long postdoc, actually probably postdoc and research associate, because he stayed there for a long time with Louis Kay. And that's probably the reason why you don't have shoulders anymore, Pramodh. And the reality is that he had a plethora of contributions on the dynamics and particularly investigating the invisible excited states by relaxation dispersion and other techniques by NMR. And there are many contributions from Pramodh with Louis there that are seminal works. Then Pramodh, after that period, moved to India, back home, where he's currently a reader. He has a position there, and he continues interested in all these studies for dynamics using NMR spectroscopy. And that's exactly what he's going to talk to us about today. So Pramodh, whenever you want, the floor is yours.
Okay, Oscar, thanks a lot for the kind introduction. And I'd like to thank all the organizers for organizing this very nice seminar series that I really look forward to every Thursday evening, and for giving me a chance to present some of the work been doing at TIFR Hyderabad. So today I'll speak about some new CEST experiments that we've worked on for the last few years to study protein conformational exchange. So the first question that may come to mind is, why worry about conformational exchange when you have this nice NMR crystallography or now cryo-EM structures of your proteins? But as we know, proteins move on a rugged free energy surface, and the structures we get usually come from this low energy conformation that's down here. And so if you're interested in folding, you want to know about folding intermediate. Similarly, the active conformation of your protein may not be the lowest energy one, but a high energy one. So to understand mechanism, you want to know something about this state. And similarly, some of these may be aggregation prone. So you want to know something about them to understand disease. So if you think about it this way, it's not enough to know one structure, but you actually want to understand the free energy surface. And so how might one go about doing this? So we can start by detecting minor conformers populated to as low as 0.5% or lower in the future. We want to understand the interconversion process, because if we get an idea of the populations, it will tell us about the free energies of the states. And if we get an idea of the lifetimes of the states, it will tell us the heights of the barriers that separate the state from other states. And we would also like to get the structures of these minor conformers. Now, often we have a dominant conformer in exchange with other minor conformers, and that's shown here for the case where state A is populated to 90% and state B to 10%. And so if we are at very low temperature essentially in the absence of exchange, we expect to see two peaks in our spectrum. and with a ratio of 9:1. But the moment we have exchange, this minor peak disappears and becomes invisible because its lifetime is 9 times lesser. And so we have to detect these other minor states by doing something to this visible major state magnetization. And so there are a couple of ways you can do it. You can use the very nice pressure jump, concentration jump, or temperature jump experiments and see how the system returns to equilibrium. Or we can also try studying it at equilibrium without perturbing the system, which is what I will speak about today. And all these methods are complementary because they will let us study different intermediates if we are lucky. Okay, so today I'll speak about the attempts to understand the folding of this protein T4 lysozyme. So it's a small 164 amino acid protein with 2 domains, the N-terminal domain and the C-terminal domain. And people have been looking into its folding mechanism for several years. And what we know is that in addition to the unfolded and folded state, there are other intermediates, which I'll call A and B for this, today's talk. So A is quite a bit different from the folded domain, while B is thought to be very similar to the folded protein with some small differences in the N-terminal domain. Okay, so the questions for us was, can we detect and characterize these folding intermediates, and can we get any insights into the folding mechanism? Is it this one where B is involved in folding, or this one where B is not involved in folding? And what we know is that the exchange rate is around 10 per second. Okay, as we know, biomolecular conformational dynamics occurs on the picosecond to hours time scale. And we can use different NMR experiments to probe dynamics occurring over different time scales. And so as protein folds at a rate of about 10 per second, we will use the chemical exchange saturation transfer or CEST experiment to study this dynamics. Okay, and so let's see what the chemical exchange saturation transfer experiment is. So here we start with magnetization along the Z-axis and apply a weak B1 field for several hundred milliseconds, and then see how much of the magnetization remains along the Z-axis. And we do this at various offsets. So suppose we apply radiation somewhere here, which is far away from both the major and minor state resonances. We don't expect anything much to happen. However, if it's coincident with the minor state resonance, spins in molecules in the minor state will undergo Rabi oscillations and precess around the B1 field. And so when the molecule jumps back to the major state, magnetization won't be along Z anymore, and this will lead to a loss in visible major state signal. So if we make a plot of the intensity of the major state signal versus the offset at which we are applying the B1 field, of course, expect to see a dip at where loss in magnetization where the major state resonates, but we'll also see a loss in magnetization where the minor state resonates. And so this way we can detect the minor state. And so this idea of chemical exchange saturation transfer has existed since 1963. And a lot of the things I'll tell you in the first part of the talk are pretty much there in this paper. So this shows a modern CEST experiment to study exchange at backbone 15N sites. And so this is basically a 15N T1 experiment with a CEST period put in here. And followed by, so basically we will get a HSQC spectrum where the intensities of the peaks will depend on the offset at which the 15N B1 field is applied. And what we also do is do 1H decoupling at this point so that it basically reduces the system to an isolated spin half. Okay, and so this is the HSQC spectrum of the FF domain, A39G mutant at 1 degree, and it's very well dispersed as you will expect for a folded protein. However, if you do the CEST experiment on this, you find that in addition to the major state dips, you also see these other dips, which tells us that the protein even at 1 degree is in exchange with a minor state. And so by analyzing these CEST profiles, using the Bloch-McConnell formalism, we can get the rates and populations of these minor states. So here we know that population is around 1.5% and the lifetime is around 20 milliseconds. And we can also get the chemical shifts of the minor state resonances, which tells us here that the minor state is unfolded. And we can also get the R2 values of these minor states from the width of these dips, which is very useful. Okay, so they have been saying things so far, it appears like it's very easy and to do the experiments and everything works all the time, but that's not always the case. So today I will tell you about this double resonance CEST experiment that came up with to study multi-state exchange. And more specialized glycine 1H alpha cyst experiment. Okay, so returning to T4 lysozyme, this is the spectrum at 50 degrees where we can study the folding. And as you can see, it's very well resolved, and we have 144 well resolved peaks. So which means we can basically study dynamics at almost every site in the molecule. And so when we went ahead and recorded CEST profiles, 15 and backbone CEST profiles, we saw that almost every peak showed at least one small dip in addition to the major state dip. And for several residues in the N-terminal domain, we saw 2 dips. And so that is illustrated here for threonine 26, where we have the major state dip and 2 minor state dips, and so on for leucine 32. So this told us that we have at least 3 states, the folded and some other states. And so we could say that we have 2 minor state dips by doing these experiments in a temperature dependent manner. And so in the first set, there are changes in shifts all over the protein. And this is illustrated here where the absolute value of the chemical shift difference between the major and minor state is plotted as a function of residue number. And you can see that the changes are as high as 6 ppm all over the protein. So we guessed it's unfolding. It's the unfolded state. And so what we found is that there is a very good correlation with what we expect upon protein unfolding. So which is nice because we have the unfolded state, and that's nice if you want to study folding. And for the second step, for the second minor state, we found that most of the changes are localized to this blue region of the N-terminal domain. And so we'll call this state B for today's talk. So we're detecting this U state and the B state. So then we said, what is the folding mechanism? Is it this linear mechanism where the unfolded state has to go via B into the folded state, or B is just an alternate conformation which has nothing to do with unfolding, or do all 3 states convert with each other, or is B a misfolded state? And so we went ahead and analyzed CEST data. And so in brown here is brown dots are the CEST intensities. And in magenta are the fits to this linear model where folding goes via B. And what you can see here is that we got very nice fits to all our data. I've shown only some here. These were the exchange parameters we got. So at this point, we were very excited because we thought we had detected this folding intermediate, and so that we could work on it. But then we decided to check if the data satisfied an alternate model where B has nothing to do with folding. And so once again, the fits are shown here in green, and as you could see that it fits very well again. So basically the CEST data is not differentiating between these 2 models. And you may think we didn't have enough data, but all the fits did here were with at least 3 B1 effective fits, and even adding 5 didn't make any difference. So we could do all this thanks to the new BCEST sequence where we can fold the spectra a lot. So then where does this leave us? We could rule out this misfolding model I didn't show you, but the fits are not good, but it doesn't tell us whether B is a folding intermediate or not. Okay, so then we went ahead and tried to figure out why regular CEST failed and see if we can tweak the experiment in some way to differentiate between these models. So, what we did was to calculate sort of idealized CEST profiles for these exchange parameters. And I've done that first here for this linear model shown in magenta, where we have the 2 minor states and the major state, and then next calculated it for this bifurcated model. And the idea would be if there are any differences between the 2, we will tweak the experimental parameters to maximize the differences and differentiate between the models. So when we calculated it for the second model, what we found is that there is almost perfect overlap. So this told me that we won't be able to do anything with the regular CEST experiment easily to differentiate between these 2 models. So then came up with the idea of this double resonance CEST experiment to study multi-state exchange. So the idea here was rather than carrying out a single frequency irradiation and see what happens to the major state resonance, we will carry out irradiation at the positions of both the minor state dips and see what happens to the major state resonance. So what was the logic behind this experiment? So our thinking was that if exchange follows this linear model, and we can carry out simultaneous irradiation at omega B and omega U. When the molecule goes from F to B, and if B1 irradiation is high enough, then it will sort of get dephased here. And so additional irradiation at U should not have any effect on the size of the dip. And so then when we did the calculations, that's exactly what the calculations told us. So if we look at the size of the dip at omega B, when we are carrying out simultaneous irradiation at U, it's not much different from what we got using regular CEST experiment. On the other hand, if we have this bifurcated process, we have two individual, two independent processes. So the thinking was, if we have simultaneous irradiation at omega B and omega U, the size of the dips will increase. And then when we did the calculations, that's exactly what we saw. The size of the dip increases when we carry out simultaneous irradiation. So the idea was we should be able to differentiate between the two processes by doing the double resonance CEST experiment. And so once again, the double resonance CEST experiment also was proposed in 1963. And these have been used to study exchange between visible states. So we went ahead and recorded double resonance CEST data at 6 sites where we have well separated major and 2 minor state resonances. And so this double resonance data is shown here. And so when we fit everything together to this linear model where folding has to go by a B, we found that this cannot satisfy this double resonance data. On the other hand, when we fit it to this bifurcated model where B has nothing to do with folding, it satisfied it very well, and we got back exactly the same exchange parameters as we had before. So this told us that T4 lysozyme can fold from U to F without going through the state B. Okay, and we confirmed this by doing more calculations and things like that, and we can say that B is not necessary for T4 lysozyme folding. Okay, so then this brought us to the question of what is the conformation of T4 lysozyme in state B. We know that the changes are localized to this region of the N-terminal domain, and state B exchanges with F at the same rate as which state U exchanges with F. So we thought maybe it's locally unfolded. But when we compared the chemical shifts of state B with what we expect upon unfolding, there was no correlation. So this told us that there is no local unfolding in B. So we got even more intrigued and said, okay, let's try to see if we can learn something about the conformation. So we had to go beyond 15N experiments, and we did all the rest of it under 2-state conditions. Okay, so we recorded carbonyl CEST experiments, C alpha CEST experiments, and C beta CEST experiments. And all these also are the changes in chemical shifts are localized to this region of the N-terminal domain. Okay, so next we had to record proton CEST experiments to get the minor state chemical shifts of protons, amide protons and also H alpha protons. But as we know, amide proton-proton NOEs will give dips in CEST profiles, which may lead you to believe that there's a minor state when there isn't one. Luckily for us, this problem has been solved earlier. There is this indirect way of doing N15 CEST experiments to get out the amide proton minor state shifts due to Guillaume-Boulny and Louis K. And also there is more recently this direct amide proton CEST experiment from which is done in a spin state selective manner to get the minor state chemical shifts. And this one is more convenient. So I'll speak about this one. And so this worked very well for amide protons. But let's just see this in the context of a CH group. Okay, so here we have our proton of interest, which resonates here, and it's attached to a C13 nucleus. And we also have another remote proton, which is attached to another C13 nucleus. And these two protons interact due to an NOE. And if you calculate the CEST profile for this proton, what you expect is 2 dips here, because we are not doing any C13 decoupling. So one corresponding to carbon in the alpha state and one corresponding to carbon in the beta state, and also 2 dips here corresponding to this carbon in the alpha or beta state. And dips here come due to the NOE interaction between these 2 protons. Now, if we follow the prescription according to Tairanio and Ashok Shekhar and Louis K, we should separate the 2 profiles based on the spin state of this C13 nucleus. So we'll separate one with C13 in alpha position. So we have only one dip here, and one with C13 in the beta state. So we have only one dip here. Okay. On the other hand, here we still have both these dips, because this spin state of this C13 is uncorrelated to the spin state of this C13. And so if you take the difference between these, the NOE-induced dip gets cancelled, while the dip due to the major state or any minor states you have will remain. And so as you can see, this doesn't even have to be C13. You don't have to have a heteronucleus here, or you don't have to have can be a 15N. Okay, and so it worked very well for protons, as we would expect, and it worked very well for non-glycine H alphas, which I have shown here again, and so we got them. When we went on to do it for glycine H alphas, where we have very well resolved spectra, what we found is that for the two geminal protons, we got the NOE-induced dip there. Okay, so this we had to get rid of this somehow. This brings us to this glycine H alpha CEST experiment. And so we now know how this experiment works. So let's see why this, repeating this blindly for the glycine H alpha sites failed. So in the case of glycine H alpha, we have two protons. This is, let's call this the proton of interest, but both of them are connected to the same C13 nucleus. So if you calculate the CEST profile for this proton, again in the absence of exchange, we see two dips here because we are not doing C13 decoupling, and same here we see two dips for this one. Okay, and they come because of the NOE between the two. And so superficially, these two look very similar to each other. Now, if we separate them according to the spin state of the C13 nucleus, so here is one where C13 is in the alpha state, and so you have only one dip here. But the difference is that we have only one dip here also, because this proton is connected to the same C13, unlike here where this proton is connected to a C13 which is uncorrelated to this one. And same thing happens when C13 is in the beta state. So when you take the difference between these two, you get the NOE-induced dip. And so this looks exactly as what we had seen before. Okay, so now we sort of understood where it came from. So how do we get rid of it? I mean, one way would be to come up with an experiment to study this in CH2 groups, but we thought others have solved it for CH groups, so let's just work with CH groups. So we just said we'll 50% random fractionally deuterate our protein. So then we'll have 25% CH2 groups, and we have to suppress the signal from these 25% HCD groups and DCH groups, which we want to detect, and 25% CD2 groups, which we cannot detect in our proton detected experiments. And so this is the experiment we came up with, and it's based on this experiment by Taran and Louis K. And so the idea is here we have a proton CEST period, and then here we have this CH2 filter, which gets rid of any signal from CH2 groups, followed by a standard HSQC part. So once again, the intensities of peaks in the HSQC will get as a function of offset, can be analyzed to detect the minor states. So if we did the experiment without the CH2 filter, we would see correlation arising from HCD groups that we would like to see, and also from CH2 groups that we don't want, but this filter suppresses these. So then when we went ahead and did the experiment on the 50% deuterated sample, this is the HSQC spectra, and it's narrower than what we got on the fully protonated sample. And also the other nice thing is these NOE-induced dips due to the geminal protons are no longer there, because there is no geminal proton. So with that, we could get our full set of H-alpha resonances in the minor state. So now we're going on to get things in the side chain and things like that. And then hopefully with all that, we will get an idea of the structure of the protein in state B. So to summarize what I've said so far, CEST could detect the unfolded state and the state B of T4 lysozyme, but it didn't tell us anything about the exchange mechanism. So we developed this new double resonance CEST experiment to study multi-state exchange, and that told us that T4 lysozyme folds directly without passing through state B. We could also say that the molecule is not locally unfolded in state B. And then we start to come up with this new experiment to obtain the minor state chemical shifts at glycine H alpha sites. Also think it's useful for other methylene groups in proteins and nucleic acids. So what are we doing now? So I didn't tell you anything about this state A, but we can infer the presence of it from our CESS profile. So we want to know if that is involved in folding or not. And also, of course, want to first know what the conformation of the protein is in state B, and hopefully in the future we'll know something about state A. And so that I'd like to thank people who did the work. And so Ved was responsible for much of this work. Shubhendu also contributed to identifying some artifacts we initially found, and also used the Hubble resonance test experiment to study conformational exchange in HPF. And Shamashri, when she was rotation student, started this work. I'd like to thank Taihan, Ranjit, and Louis for sharing pulse sequences and lots of suggestions. So I'd like to thank Krishna Rao and TFR and MR Center for lots and lots of help and spectrometer time, some funds from SERB, and lots of intramural funds from TFR for everything from chemicals to spectrometers. And I'd like to thank all of you for your attention.
Thank you very much, Pramodh, for this very educational, interesting seminar. So there are a couple of questions, but I have first a question. In this, when you try to unravel, I mean, you use CESS beautifully to distinguish between the two different steps in the energy landscape. But have you considered using for co-solutes, for instance, a slow amount of denaturients, because perhaps they could differentiate, and then you could see that by CESS directly and not by double CESS? Is that meaningful to you, or?
So we tried initially. Actually, we tried a lot of things, adding different like varying salt, pH, adding urea, and those things. We didn't succeed in differentiating between the mechanisms.
Okay.
So that's when we came up with this idea.
Yeah, well, yeah, sure. Okay. So there are a couple of questions at least. Orlowski asked if there is a way to apply these techniques for solid state NMR. I believe he asked that while you were describing the CESS techniques.
I don't know much about solid state NMR. My colleagues here do a lot of these, but yeah, I have no idea if they can be applied, and how it will interfere with the spinning, and if it coincides with the sidebands or something like that.
Certainly for imaging, because I believe that these things come from imaging, where parasites. Yeah. But I don't know either. And there is Supriya Pratihar asks, when you have the major visible peak in between two minor peaks invisible in the CESS profile, isn't it obvious that the third state have to be off pathway, since the chemical shift of the third state is not between the folded and the unfolded?
No such thing. I mean, if you, okay, I showed two profiles here, but I mean, so what people thought at least is you have the unfolded state, which is very different, and B is very similar to F, and folding has to go via B. And if you see a lot of the places, omega B is pretty close to omega F. So it won't be so straightforward to rule out, say, just from the chemical shifts that B is not involved in folding. If anything, from the chemical shifts, you would almost guess that B is involved in folding.
Okay. So then anonymous attendee asks, how do you pulse on two different frequencies at the same time?
Okay. So I have a slide for that. Okay, so there are two ways we could have done it. One was to use, during the B1 irradiation, use cosine Morpheus modulated pulses, and that would probably be very nice and probably the way to go. But the only negative thing about it is just while fitting, you have to either say you have a time dependent field and one stationary field, and you can do that, or the other way is to say, you calculate a Liouvillean for each of these small rectangles, and so then you can calculate the evolution of the density matrix. So everything will be fine, but fitting will be cumbersome. Like rather than take a few minutes, it could take a few hours. On the other hand, so that's why we used this DCEST experiment where use the Dante train to do double resonance excitation. So we put the carrier on one state and adjust the delay between the two pulses to the frequency difference between the two, 1 over the frequency difference between the two. And in that case, we have to calculate only two propagators, one for the delay and one for the pulse. Then if we exponentiate it, we can calculate what the CEST period does. So we did double resonance irradiation using this Dante train.
Yeah, I guess this is very related to the question that Jeffrey Van Housen had, which is actually he asked about how did you generate the two frequencies, but he also said whether it does, for double resonances, does it help to have distinct radio frequency amplitudes at the two frequencies?
So yeah, we did consider using distinct radio frequency amplitudes, but reason we didn't really proceed with that here is, let's go back. The populations and lifetimes of the two states are quite similar. So I think if the lifetimes of the two states are somewhat different, it will be better to use different radio frequency amplitudes for the two frequencies.
And anonymous attendee asks an almost topological question. So asks whether it's possible to roll out the triangle exchange model by running double resonance CEST experiments.
Okay, so I don't want to say we can rule it out, but when we fit the data to the triangular model, what we find is the minimum comes when this thing KEX BU is 0. So what we could say for sure is this pathway does not have a big role, and we can say for sure B is not necessary for folding, and we can also say that this pathway that goes via B does not have a big role, and the minimum comes when KEX BU is close to 0, but I don't want to say we can rule it out or anything like that.
Mm-hmm. Another anonymous question asks whether you can comment on NOE cross peak to water.
Okay. 1 second. Sorry. Yeah, so in this proton CEST experiments, at least the main worry is that, if there is another proton that resonates in this region, you will see a dip due to that, and you will, you will attribute that to a minor state. In the case of amide protons, water is first of all, to some extent it should get subtracted out, but anyway, water is very far away. So you will never think the amide is resonating on water. And if that happens, you'll probably you'll be just careful and ignore it. In the case of H alpha, in our case, we did the experiments in D2O. So we didn't face that problem.
Oscar, can I ask a question?
Definitely, you have this privilege.
So, of course, first two comments. Thank you very much for referencing the Allerhand and Thiel paper. I recommend it highly to graduate students and postdocs. It's a terrific paper. And also for reminding me of Mark Rantz's work. I'd forgotten about that paper, so I'll go reread it this afternoon. My question is, the folding rates for lysozyme seem remarkably slow. And how does, under your conditions, how do those match to other techniques for measuring folding? And more generally, how fast a process will the CEST experiment work on?
Okay, so so I've forgotten now what other stop flow experiments measured, but I think these compare quite well because we chose conditions that people had sort of close to what people did stop flow under. Regular CEST may work easily up to 500, 600 per second, but double resonance CEST will work well for slower processes because when you carry out irradiation, you really have to be able to carry out irradiation that is fast compared to strong compared to the lifetimes of these states, and in which case if V1 becomes very high, it will start touching multiple states, and so it won't be very useful. So I think 100 per second is where double resonance will hit a limit, or maybe even a little lesser.
Okay, so I think that this is another question by Hari Babu, which is actually, it's related. What is the practical limit of the two frequencies in terms of separation between them? But, so in the Dante, essentially, what is the resolution of the Dante, I believe?
No, so the thing is, what we found is, in hindsight, it shouldn't have been surprising, but for this double resonance to be useful, B1 has to be strong enough sort of to dephase magnetization. Okay, so you can't use small B1s and study peaks that are close to each other. So that will put a limit. And so folding is very nice because things are well separated.
Mm.
But I've studied other cases also where things are not but not so well separated. But one nice thing, I didn't show this slide, so I can show this now. One nice thing is what we found here is that so each of the 6 sites that we had exchange double resonance S data for, we could fit it to the exchange process. And so the results for each of these sites is shown. And surprisingly enough, all of them agreed very well with each other. So you could at least get some insights by studying one site where things are well resolved.
I see. So Emmanuel Berutis asks, what is the advantage of CEST over STD in this context? I not fully understand.
No, I think of both, at least I'm not very sure the answer, but at least I think of both as almost the same experiment. But yeah, so I can't exactly answer what the advantage is.
Okay. Anonymous question, do you already have an idea about the secondary structure of state B just from chemical shifts?
Yeah, we have an idea. We know the backbone. So if we put, if you take all the chemical shifts and put it in Talos or the program, what you will see is that conformation of state B is almost the same as the folded state. So if there are changes in the backbone happening, it's very subtle. Despite these rather large N15 chemical shift changes. So could be at the level of side chains. That's what we're checking now.
Okay. That's probably why it didn't work, the urea, because they are very similar.
Yeah, only the unfolded state population changed.
Oh, for sure. Yes. And it is possible to apply similar random scrambling technique to study side chain NH2 groups.
Possibly, maybe you just have 50% D2 or something like that as the solvent. Could work. Okay,
that was the question. I mean, there were many questions for both Ani and Pramodh that actually reflects the interest that this seminar series actually raised among the attendants, which was very high, by the way. So I would like to congratulate you both for these excellent seminars. And I think that probably we should stop here the official part of the session. Just simply reminding that next week we'll have another meeting here with Bipin Agarwal and somebody else who will yet to be confirmed. But we encourage you to also attend. And now I think we can continue in a more informal way.